Welcome to How Cellular Respiration Works by Haven, Chad, Evan, and Jack. We are the support team.
Glycolysis means sugar splitting. During glycolysis, glucose, a 6-carbon sugar, is split into 2 3-carbon sugars. Then, the smaller sugars are oxidized and the remaining atoms are rearranged to form pyruvate. Glycolysis has 2 phases, energy investment and energy payoff. During the energy investment stage, the cell spins ATP, and this investment is repaid with interest during the payoff stage. During the payoff stage, the ATP is produced by substrate-level phosphorylation. NAD+ is reduced to NADH by electrons, which are released by the oxidation of glucose. The net yield per glucose molecule is 2 ATP plus 2 NADH.
Pyruvate oxidation is when a pyruvate is converted to acyl CoA. It also releases a molecule of CO2 and reduces an NAD+ to NADH. A pyruvate is 3 different carbons linked together. First, a carboxyl group is removed from the pyruvate and is released as carbon dioxide, or CO2. Then, the carbon molecule is oxidized, and NAD+ accepts the excess electrons to form NADH. The oxidized carbon molecule is then attached to the coenzyme A to form acyl CoA.
The citric acid cycle functions as a metabolic furnace that oxidizes organic fuel derived from pyruvate. The cycle starts by the acyl CoA joining the cycle and combining with the compound oxaloacetate. Next, 2 completely iodized forms of CO2 leave the cycle. Throughout the cycle, 3 NAD+ are reduced to 3 NADH molecules. In every turn of the cycle, 1 ATP is created by an ADP molecule added with a proton. Then, FAD, which accepts 2 electrons and 2 protons, becomes FADH2. Through the steps of the cycle, citrate is decomposed back to oxalate, which is then regenerated, making this process a cycle.
Oxidation phosphorylation, also known as the electron transport chain, is a process in which ATP is formed as a result of the transfer of electrons from NADH or FADH2 to oxygen, and the final step of cellular respiration. It takes place in the inner membrane of the mitochondria. Involved in the process is protein complexes 1, 2, 3, and 4, and the ATP synthase. First, NADH is oxidized to NAD+, leaving behind 2 electrons, which are passed along the complexes. Next, the electrons pump out hydrogen ions. Then, FADH2 is oxidized, releasing electrons to form FAD, which creates protons that combine with oxygen to form water, or H2O. Lastly, the hydrogen ions go back into the membrane through the ATP synthase, which creates gradient, and form ATP.
The next step of cellular respiration is fermentation. Fermentation is when your body isn't receiving enough oxygen, so it resorts to converting carbohydrates into alcohols/acids to obtain energy by using yeast and bacteria. Fermentation is anaerobic, but it is not anaerobic respiration. It starts off with glycolysis. Cells break down glucose into 2 ATP and 2 pyruvate and 2 NADH. The pyruvate stay in the cytoplasm and are used to produce lactic acid and/or ethanol. NADH plus from glycolysis is recycled back to NAD plus so glycolysis can continue. Fermentation allows glucose to be continuously broken down to make ATP due to the recycling of NADH to NADA plus. The example that I'm showing here is lactic acid fermentation, which only humans can do. An example of that would be running. Yeast cells do alcoholic fermentation, and an example of that would be beer.